Okay, we are gonna get started, continuing our journey through the cell with chapter 6. Just a couple reminders, there's gonna be a quiz on Moodle on Friday. That's gonna be on everything that we have covered up through today. So whatever we get done today through the cell is fair game. I don't know how far we'll get today, so the quiz isn't done yet, but when we finish today, that's the end of what will be on the quiz. Your next test, I'm just giving you a heads up. Looking at the schedule, the next test is gonna be Wednesday, October 2nd. It's on chapter 6, 7, and 8. So 3 chapters, so chapter 6, we will finish on Friday, maybe starting chapter 7. Next week, we'll definitely have time to finish 7 and 8. So the test will be on Wednesday, October 2nd. So remember on our syllabus, it just says what week it is. So I'm just letting you know it's gonna be Wednesday instead of Friday. Your test that you took last week is gonna be back in your folder this afternoon. So just when you get it back, it will say a bunch of numbers on it. The test was out of 57 points, so that'll be on there, and then there's gonna be a couple different numbers. So W is your homework average, Q is your quiz average, E is your exam average, which obviously there's only one of those, and then O is your overall average at this point. So that's incorporating everything into what your overall average is. The test average is around a 60, 61. That is normal. It's not fun to hear, but that is normal for a first test. So I just want to encourage you with the idea of that does not make, or this test, one test does not make or break you. Okay, one test is not going to, one poor test grade does not necessarily mean you're not going to make it, and one great test grade doesn't mean you're getting an A either. So there's 5 tests. We have 5 tests plus a final. So we have 5 in-class tests plus a final. So this is not a do or die kind of test. Okay, that's another reason that I want to give you your full overall average when you look at it, is because if you didn't do well on the test, but you're doing well on homeworks and quizzes, it's going to show you that that's, again, it's not the only thing. If you did well on the test and haven't turned in homeworks or done quizzes, that also has an effect on your grade, but giving you an idea kind of where you stand overall. This is not a final grade, and there's a lot of time for that to change, and there's a lot of assignments for that to change. Okay, so you will get it back on Friday. I'm thinking we are going to have a little bit of time. We may go over some of the tests, so bring the test with you to class on Friday. Okay.
What about the people that had lab yesterday?
It'll still be there. Yeah. Yes. So I'm gonna put it in after lab today. I know that you want to see it as soon as possible, but if I put it in before lab, that's all that you will think about during lab. I've learned some things in my life. So I talked to Ashley and I was like, I'm not putting them until after lab. So I'll put them in at the end of lab today. So if you had lab yesterday, you can stop at the lab anytime. You have swipe access to that lab. The lab locks at either 6 or 7. I don't remember, but you will have swipe access to that. So you can get in anytime and they will be there anytime. And if you have lab today, you could grab it at the end of lab. And if you have lab on Thursday, you can wait till tomorrow if you want. Maybe you're one of those people that doesn't want to know and will stand by the folders for 20 minutes looking at it and saying, I don't want to look, but I do want to know, and make somebody else look at it. That's up to you. Okay. It'll be there this afternoon. I do want to talk about some study ideas and just remind you of a couple things, and we'll talk about these again as we approach the next test. But you may remember at the beginning, we talked about there's 3 things that you need to do to be successful in this class. One is preparation. So I want you to take a minute right now and think about what is one thing that you're doing in preparation for class and one thing that you can do. A lot of, we had a faculty retreat this, before the year, we always have these workshops, and we talked about the fact that you need to have a concrete plan. So I want you now, Ed, to take a minute to write this down, because writing it down makes it more concrete. What is something that you are doing to prepare for class? And what is something else that you could do in the future that will help you be more prepared for class? Another thing that helps you with this, follow through on these things, is telling someone else this and having them hold you accountable, but I'm not gonna make you do that. Okay, just as a reminder, homeworks, the idea of homeworks isn't their preparation, that they're helping to prepare you. What are some other things, anybody have any ideas you want to share? Things that you can do, prepare for class? What? Read the book. Which, again, the homework is trying to force you to do that, but I recognize you can do the homework without reading the book stuff. But reading the book, not a bad idea. You paid for it, so use that resource. Highlight in the book, underline in the book, take notes from the book. Okay, the second thing I said that you needed to do was be attentive in class, being active in class. So again, take a minute to write down something that you are doing in class that you feel like you're doing well. And then something else that you think maybe you could do to help you be more attentive in class or more active in class. Putting away your phone would be one. And then the third thing I said that would help is reviewing. So after class, at some point, doing something to review material. So think about something that you are doing. Quizzes are one of the things that are supposed to help you review. What are some other things that you could do to help you review? And again, the more concrete and specific you are about how you're actually gonna do this, the more likely you are to actually do it. So a lot of people on your test, you talked about that you were going to review your notes after every class. That's a great idea. So make yourself a plan and say on Monday, Wednesday, and Friday at 9:30 p.m., I'm gonna take half an hour. And put it in your schedule and tell someone you're gonna do it and do it. Another thing that you can do to review, and several people wrote about this on your test too. At the end of in the book, at the end of each section, there are questions. And the answers to those questions are in the back of the book, so you can answer those questions and then check your answers. There are also questions at the end of the chapter overall that you can use to help you review. If you have the e-text, I'm guessing there's lots of stuff in that too. I honestly don't look at the e-text ever, but I'm guessing the same kinds of things are available, if not more. Flashcards, I can't say it enough, flashcards. My daughter's in 9th grade. She's in tears last night, just in the first 30 elements of the periodic table. Crying. I was like, why don't you make some flashcards? So I made her sit down with 3 colors and the next cards, and she made flashcards. I thought she had memorized like atomic number and all that. She just has to know the names and symbols. I was like, you can do this. You'll be okay. I thought she had to know them all in order but I know that it's old fashioned and Quizlet, it works and that kind of thing works too, but I still believe that writing things down makes a difference, which is why I want you to write these things down and think about these things. Okay. So we left off on Monday. We talked about the cell membrane, we talked about the nucleus, the nucleolus, and we talked about ribosomes, and that's kind of where we left off in the cell. We talked about the difference between prokaryotic and eukaryotic cells. So we're focusing on eukaryotic cells for the rest of the chapter. And we're going to keep going further into the cell. We're looking at the group of membrane-bound organelles that are referred to as the endomembrane system. Okay, so the jobs of this system. So this is a group of organelles all kind of working together. They regulate protein traffic, and they have a lot to do with metabolic functions. So the endomembrane system is a bunch of organelles that are working together, regulating the protein traffic and a lot of different metabolic functions. And we're going to go more specific than that, but this includes the nuclear membrane, which we talked about, the nuclear envelope. the endoplasmic reticulum, which we will abbreviate as ER, not to be confused with the emergency room. This comes in two forms. The rough ER and the smooth ER. Reticulum. It includes the Golgi apparatus. Sometimes called the Golgi body. My high school biology teacher had a parakeet named Golgi. That's how I remember it. Lysosomes, that's an E. I put two S's, I don't know why. And the vacuoles and vesicles. So these different organelles are working together to make proteins. When we talk about protein trafficking, we're talking about making proteins, transporting those proteins, and then metabolic functions, things like cellular respiration, moving lipids around, detoxifying poisons, that kind of thing. So they all either are touching each other or connected to each other, or they can move things around using vesicles. So they have direct contact, or they move things around using vesicles. And actually, we should actually add the plasma membrane itself too. So it's kind of a linear flow when we're talking about proteins, like making proteins, moving them through the endoplasmic reticulum, through vesicles to the Golgi body, through more vesicles, it can go all the way outside the cell if we're talking about secretory proteins. So if we go back to our diagram, let's label these guys before we talk about them. So we already labeled the nucleus and all that stuff. We don't need to relabel that. Where is the endoplasmic reticulum located in relationship to the nucleus? Where is the endoplasmic reticulum located, CJ? So yeah, it's all this blue stuff that's surrounding the nucleus. I know that sometimes these arrows are a little bit tricky. This is the diagram that's in your book. I tried to use the same one because some organelles are easy to identify based on how they look, and some of them are just little circles, so they're hard to tell. So we have endoplasmic reticulum. We have both the rough ER and the smooth ER. And again, on this diagram, they're both blue. If you're one of those people that finds it easier to correlate colors with things, print the diagram out and color it. If that helps you associate a color with something, location, if that can help you. So this is smooth ER, rough ER. What's the difference between smooth ER and rough ER? Yeah, the difference is the ribosomes. So the rough ER has ribosomes that are stuck to it. So that's all those little purpley dots on the outside. And the smooth ER does not have the dots, the ribosomes stuck to it. And they have different jobs, which we'll talk about in a minute. Okay, so we've got rough ER, smooth ER, what it would be another part of the endomembrane system that we just listed. The Golgi apparatus. So what color is that on this diagram? It's kind of this like tealy bluish color. And then again, the other ones that are not particularly distinctive in their structure, I'm making sure I get the right line. This pinkish one is a lysosome. And there aren't really any specific vesicles on there, so we'll talk about them and where they are and why they're not necessary. Like this actually probably is a vesicle. This is a vesicle. coming off of the Golgi apparatus. Okay, so let's go back to, sorry, we gotta go through everything. Endoplasmic reticulum. So endoplasmic reticulum takes up a lot of space in the cell. In lab this week, you're looking at these cell models, and I don't feel like we should do justice to maybe how much is taken up by the endoplasmic reticulum, but again, you can see in these cell models, the endoplasmic reticulum is also blue. It's not like it's blue, but it's not really blue itself, but we apparently always show it as blue in our models and diagrams. So endoplasmic reticulum is a huge organelle. It can take up to about half of cell membranes are endoplasmic reticulum. So it's a lot. It's connected. Can't spell today. It's connected to the nuclear envelope, so it's continuous with it. So it's directly connected to the nucleus. Endoplasmic, endo means within, and plasma is referring to the cytoplasm, so it's within the cytoplasm. And reticulum, I think I misspoke on Monday. I think I said that lamina meant little net. I was thinking of this. Reticulum means little net. And its overall function is it's a factory. It makes stuff. So it's a biosynthetic. Meaning it's making molecules that are really important for living things, macromolecules that are important. So it's a factory. It makes stuff. And again, we're going to divide it into the two categories because they have different functions. On this diagram, again, you can see the difference. We've got the smooth ER here that doesn't have the ribosomes on the outside. And then this is actually the rough ER with the ribosomes on the outside. And this is showing you how it's continuous with the nuclear envelope. So connected to the nuclear envelope. So we've got all this membranous, all these tunnels, cisternae. These then can bud off and form these vesicles, which can be used to transport the things that the ER is making. It's a factory that's making stuff, and then it's got to transport that stuff to other places in the cell. And it does that by just pinching off a little vesicle, which is containing whatever protein or whatever is going somewhere else, and taking it to another location. Inside the inside space of this doesn't show up very well, but the inside space is called the lumen of the endoplasmic reticulum, or the cisternae, which is CISTERNAE. I don't know. I guess it's kind of big enough there. Maybe, maybe not. Okay, so let's talk about, you want to start with rough or smooth? Smooth, okay. What does the smooth ER do versus the rough ER? So smooth ER, one of its jobs is to make lipids. Why are lipids so important to a cell? What do they make up? What part of the cell is made up of a lot of lipids?
Any membrane.
Yeah, so any membrane. So smooth ER are making the membrane lipids. What kind of lipids are in the membrane? Phospholipids. So remember, those are the ones that have the phosphate group and then the two little hydrocarbon tails. One of them is straight and one of them has a little kink in it. So it's not just limited. Oh, shoot. I didn't mean to do that. Sorry. I knew that was going to happen, but I didn't want it yet. So when we talk about lipids, phospholipids for membranes are one of the kinds. They also are making oils, steroids, which could be used for like Also for the membrane stability. Cholesterol is used for that. And then also some hormones are made by the smooth ER. The sex hormones like testosterone and estrogen, and also some of the adrenal hormones, which the adrenal glands are right above your kidneys. So the smoothie are making lipids with lots of different functions. Another job of the smoothie are, anyone? Or you just want me to tell you because it's the 4th week and everyone's tired now. For real tired. The smoothie are also helps with detoxification of drugs and poisons. It does this by adding hydroxyl groups to them. It can actually get to the point where if someone is taking a lot of drugs, and when I say drugs, I don't mean necessarily illegal drugs, I just mean drugs. So like helpful drugs also, that need to be detoxified. When you start to take drugs, sometimes they become less effective over time because your smooth ER are getting more effective at detoxifying. So you have to take more and more and more, and you can actually get cells that build up huge amounts of smooth ER, especially in the liver, and that can cause damage to the liver. Things like cirrhosis of the liver, which is caused by drinking too much alcohol, is caused partly because the cells are building up huge amounts of smooth ER to be able to detoxify those drugs. And then the third job of the smooth ER is it stores calcium ions. Calcium ions are really important for things like contracting cells. So muscle cells that need to contract regularly. Or they might need to be cells that are used, or calcium ion side that are used in making proteins. So secretory proteins, meaning cells make these proteins and then they secrete them to be used elsewhere. So calcium ions might need to be made for those proteins. And that's the job of the smooth ER. So again, these jobs, the smooth ER might be doing more of one thing than the other in a different cell, depending on what kind of cell it is. On the other side, when we talk about rough ER, the smooth ER makes lipids, the rough ER is covered with ribosomes, so what is it going to make? Proteins, yeah. So it's really pivotal in making proteins because those ribosomes are bound to it. So they're made actually by the ribosome. The ribosomes make those polypeptide chains. When we make a polypeptide chain, what level of structure is that of a protein? The amino acid sequence that makes a polypeptide is the primary structure, is the amino acid sequence. So that polypeptide then, so these polypeptides, if I could write the word polypeptide, that'd be great. Go from the ribosomes into the lumen of the ER, and that's where they start folding and twisting. And that's where they get their secondary and tertiary structures, and quaternary if they're going to be quaternary. So the ribosomes are making that primary structure polypeptide sequence. The polypeptides then are going into the lumen of the ER where they're starting to fold and twist and become helixes and side chain interactions. All that's happening inside the lumen of the ER. After those proteins are all made, also in the rough ER, sometimes, depending on the function of the protein, the endoplasmic reticulum also attaches carbohydrates to them. And this makes them what we call glycoproteins. Glyco should remind you of glucose or glycosidic linkage. And again, that's going to alter the structure of the protein some. It's going to alter the function of the protein. So when we attach these carbohydrates to the proteins, we have these glycoproteins. That's a covalent bond. That's an L, not a T. There are enzymes that are in the membrane, so proteins that are embedded in the membrane of the endoplasmic reticulum that are going to facilitate the attachment of those carbohydrates. We're going to talk about lots and lots of chemical reactions over the next lots and lots of chapters. And every time we talk about a chemical reaction, almost every chemical reaction we talk about in a cell is facilitated by a specific enzyme. So it's a complicated process. We're not going to name all the enzymes, but there are enzymes in this membrane that are allowing those carbohydrates to be attached. Once you make a glycoprotein, that glycoprotein then is going to be transported to its final destination, where it's going to be used by a vesicle, a transport vesicle. So it's got a specific purpose. It's going to be a protein, a specific protein with specific carbohydrates attached to it that are specific to its function, and it needs to go to a specific place in the cell, and that's the vesicles are going to take it there. And then the rough ER also has some functions in making membrane. So making the phospholipid bilayer. Again, remembering that structure, which we'll talk about more in chapter 7, it's a phospholipid bilayer, but it also has proteins embedded in it. So making those specific membrane proteins. And then also, it does also help out with making phospholipids. And again, those membrane parts are going to be transported to other parts of the endomembrane system to be incorporated into them by vesicles. So things are made in the ER, whether it's smooth ER or rough ER, they go into the lumen, they're processed if they need to be processed, they might have carbohydrates attached or whatever, and then they're going to be transported by these vesicles to wherever they need to go. Whether they're a membrane part or a glycoprotein, whatever, they have a specific destination and the vesicle is what's going to take it there. So it's being made there and then transported somewhere else by the vesicles. Does that make sense? So when you think of the endoplasmic reticulum, what are you thinking about it doing? Making proteins and lipids and transporting them, getting them ready to be transported somewhere else. Okay, so we're going to work our way outward from the nucleus. Oh, sorry, this is a picture that I wrote all over. This is showing you rough ER on the right side. This is supposed to be a pointer too, but I haven't figured that out yet. So this is showing you rough ER. There we go. So there's the endoplasmic reticulum. You can see it's a double layer membrane. And then all these little dots are the ribosomes that are stuck to the outside. Over here, this is smooth ER. You don't see the double membrane necessarily clearly, and you're not seeing the ribosomes attached to it. But the smooth ER and rough ER are usually attached to each other. How do I make it stop doing that? Okay, we'll go back to pen. Okay. So this is showing you, again, we're moving our way out. We're looking at this one that was kind of teal colored, which is the Golgi apparatus, still part of the endomembrane system. Okay, so you can see here's the endoplasmic reticulum hanging out around the nucleus. We're moving outward. in the cell, closer to the cell membrane. This is the Golgi apparatus as we zoom in. It has two faces, meaning two sides to it. So the cis face is on the same side as the endoplasmic reticulum. So this is closer to the endoplasmic reticulum. On the other side, the trans face is on the opposite side of the endoplasmic reticulum. So this is closer to the plasma membrane. This you can think of as the shipping side. And this is the receiving side. If we use our analogies of the ER being a factory, you can think of the Golgi apparatus as being the warehouse. It's a macromolecule warehouse. So it's got a receiving side because it's receiving things from the endoplasmic reticulum. What happens in a warehouse? Or I assume what happens in the warehouse. So we just built a giant warehouse in Greece, an Amazon warehouse. What's happening there? Amazon is shipping lots of stuff. What do they do with the stuff when they get it? They're gonna put it on a shelf until someone orders it. And when someone orders it, they're gonna put it in a package. Hopefully, they're gonna put the right name and address on it, and then they're going to put it on a truck and send it where it's supposed to go, right? Okay, that's what the Golgi is doing. The Golgi is the Amazon warehouse. These are the little Amazon trucks, okay? It's taking materials from the endoplasmic reticulum, and it's going to receive them. So it receives from the endoplasmic reticulum. So it receives a shipment. It might modify that shipment. It might make some changes to the things that it receives, to the molecules it receives. It can actually do a little bit of manufacturing too, which makes it not a perfect analogy, as they never are anyway. It sorts those materials, so it organizes them just like you would in a warehouse. When the Amazon tractor trailer pulls into the warehouse, they don't just kind of dump it all on the floor and just wait until someone orders something. Okay, everything is organized so they know where to find it when they need it. And then it ships those materials to where it's supposed to go. So receiving the materials, it's going to receive the materials from a vesicle. So these little vesicles are coming in to the ER to the Golgi from the ER. So they're containing something that somebody's going to order at some point. Something in the cell is going to need at some point. And that vesicle is actually going to merge with the Golgi on the cis face. So it comes in, and then the membranes fuse so that the contents of that vesicle are now in the lumen of the Golgi apparatus. So once it's in there, it might, when we put on, we're going to put sorting and modifying together, I guess. It might modify the carbohydrates that are on the glycoprotein. So the endoplasmic reticulum has stuck a carbohydrate to it using a covalent bond. It sends it on, it comes to the warehouse, and they're like, yeah, you know that you didn't quite get that right. We're going to alter it a little bit. We're going to change it. Or it might further process the phospholipids to get them, again, getting them more ready to do their job. When we talk about manufacturing, it actually makes polysaccharides. What do you make polysaccharides out of? monosaccharides. So it might receive monosaccharides, and then it's going to form what kind of links to put them together? What kind of bonds hold monosaccharides together in a polysaccharide? The glycosidic linkages. So it's gonna process those monosaccharides and modify them, making them into polysaccharides. And then for shipping, it's gonna add the tags. Okay, literally not, it's not like writing a little thing that says, send this over to muscle cell number 4 in the biceps. Not like that. We're talking about chemical tags. So this is adding tags that we're meaning like functional groups, the molecular groups, maybe like a phosphate group that says, hey, this goes over there. So those tags are specific to some kind of receptor so that that molecule knows where to go. So they're adding the tags to these molecules so that molecule is going to end up in the right destination. Just like Amazon puts 17 labels on your package and then puts it at your door and then takes a picture of it while you're standing there, taking the package from them. That's what's going on. Okay, the Golgi is putting these tags on so that the molecules that have been modified and processed are going to the right location. And again, this is showing you an electron microscope, transmission electron microscope picture of the Golgi. And you can see there are vesicles coming in. These are vesicles that are merging with the Golgi body. And then it's forming new vesicles to send it off. So this is kind of like it's called a cis trans maturation process. So molecules come in on the cis side, they are processed, modified, and packaged to be shipped off. And that's a maturation process as it moves from the cis side, the receiving side, to the shipping side. Coming in a vesicle, and it's going to leave in a vesicle to go where it needs to go. Sometimes you'll notice in this diagram, this one's going back this way. So these molecules have been modified by the Golgi, and they're still going back to the ER because they're part of the ER. The ER needs it for something else, but it still needs that processing that's done by the Golgi. So it's directional in the way that the molecules move. It's not a random process, it's direction. You good? Okay. The last part of the endomembrane system that we're going to talk about today are the lysosomes and the vacuoles. And I want to put next to vacuoles, vesicles, kind of use those terms interchangeably. We're still in the endomembrane system. Lysosomes, do you remember when we talked about hydrolysis? Hydrolysis reactions do what? They're breaking things down. Lysosomes are doing the same thing. So these are specialized compartments, again, membrane-bound compartments that are providing an environment for digestion to happen. So we'll start by talking about lysosomes. So again, membrane-bound. And it's full of enzymes that are hydrolytic enzymes. And again, hydrolytic we know means to break down. So breaking down those macromolecules that we talked about in chapter 5. Breaking down proteins into amino acids. Breaking down polysaccharides into monosaccharides. Breaking down lipids into fatty acids and glycerol. Why can't I remember the fourth one? Proteins, lipids, nucleic acids. Thank you. Breaking down nucleic acids into nucleotides. Thank you. These have an acidic environment that are necessary for the enzymes. Again, thinking about the fact that having membranes around enclosing things allows them to have a different environment than what's around them. So the pH is lower than it would be in the cytoplasm because it needs that lower pH in order for the enzymes that are there to work. Just like your stomach functions at a very low pH. The enzymes that are in your stomach function at a very, very low pH. Your intestines function at a high pH, relatively high pH, because the enzymes that are working in your intestines need to be at a high pH. So creating a specific environment that is necessary for the function of that organelle. These are made by the rough ER. These enzymes are made by the rough ER. And then they're transported to the Golgi, and then they're transported to the lysosomes. Your book talks about two different processes. One is phagocytosis, which we'll talk more about next chapter, but that's the idea of bringing in large molecules to be digested. So the two processes that can be carried out by lysosomes are phagocytosis, which again, we'll talk about next chapter. This is to digest large particles. This is how amoebas eat, by engulfing and bringing them in, and then a lysosome fuses with it. The other one is what we see over here. That's a 2 that I didn't do very well with. This one, phagocytosis, digest large particles. So the other one is autophagy. And that's what this picture is showing you, how a lysosome is involved in that. What does auto mean? Self. Okay, so this is self. And phase is to like consume or eat, because that's shorter to write than consume. So this is a way that cells can use a lysosome to break down something, an old organelle or something that's not working. This is a way to recycle parts. So we see here, here's a vesicle that has a peroxisome, which we haven't talked about yet, and a mitochondria, which we also haven't talked about yet, but these are old organelles. Your cell, if something is damaged with those organelles, doesn't want to just kind of get rid of it and waste those parts. So the lysosome comes along and it fuses with this vesicle, which has the damaged organelles in it. And then these enzymes are going to break down those organelles so that these raw materials can be used again by the cell. So lysosomes, if your lysosomes aren't working, if someone has, there are genetic disorders where lysosomes don't work, those lysosomes just fill up with stuff, and the cells fill up with stuff that can't be gotten rid of, and it obviously impairs function. If you can't get rid of waste or old worn out organelles, then you have a problem, and a significant problem. It's not a very common, they're not common genetic disorders, but if you took genetics or talked about genetics in high school, you may remember talking about Tay-Sachs disease. We won't get into it this semester. You might talk about it when you take genetics, but that's a disorder where the lysosomes are not working properly, and it impairs brain function because it can't break stuff down because the materials are undigested. Okay, then vacuoles, I don't really have space to write, but we're going to put vacuoles down here. We'll draw a line here. So vacuoles are just basically large vesicles. So they are just membrane bound sacs, again, that can be used for different kinds of things. your book refers to them as maintenance compartments. They are, again, made by the Golgi and the ER. They have a selective membrane. So they are membrane bound. So again, the environment that's inside them is different than the cytoplasm. So there's some different kinds of vacuoles that are important. There are vacuoles that are used to store or process food, and we call them food vacuoles. Sometimes the names make sense, you know? Some cells like paramecium did you guys see paramecium at all yesterday in lab, anybody? You looked at protists, right? A lot of aquatic one-celled organisms have a specific kind of vacuole called a contractile vacuole. They're there because of the concentration of solutes inside and outside the cell, or the concentration of water inside and outside the cell. They will help to pump water out of the cell to keep the cell from exploding. It's kind of important. If you're one cell and you're full of water and you explode, not the best outcome. There are also storage vacuoles that are specific to a specific kind of molecule. Like in seeds, there are storage vacuoles in the cells that will store a protein that help the developing plant grow. And then the most famous vacuole are the vacuoles that are found in plant cells. The central vacuole, that's this huge one inside this plant cell. It's a huge one you see in that plant cell model. It contains what we call cell sap, which is a lot of water, but it's also got ions like potassium and calcium. And again, storing all of those things, keeping the cell the right shape, which you did not see yesterday. You talked about cytoplasmic streaming in lab, but you are not doing osmosis stuff yet. So that is the end of the endomembrane system. So ER, Golgi, lysosomes, vacuoles. On Friday, we're going to maybe finish the cell. We'll talk about mitochondria, chloroplasts, and the other ones, peroxisomes, cell skeleton.